you can see it and that I can still see the chat window in case anything does come up. Okay, can you guys see that?
Yeah, it looks good on our end.
Okay, super. Awesome. Well, I'm going to talk to you today about a system where a lot of my research is based in the Mara Serengeti system of East Africa. And I thought it would be a good way to start by taking you all for a hot air balloon flight over the Mara Serengeti, actually courtesy of Mike, who's here today on the Zoom call as well, and who flew me over this beautiful stretch of the migration in the southern part of the Mara back in 2017. And as we fly over this kind of grassland landscape, think about, if I asked you to tell me like what drives productivity in this ecosystem, your answer would probably depend on your research background, but very reasonable answers would be rainfall, and rainfall indeed is really important, soil nutrient levels and fertility. And some of you, as particularly at this university in this program, might say animals and see the migrating animals may have an influence on grassland productivity in the system. And a smaller proportion might even say, I don't see them, but I know that there are predators out there somewhere on this landscape. And I bet that those predators could even have a potentially outsized impact on production in the system. But that is not a typical view of ecosystem productivity. Generally, in the kind of a history of ecosystem ecology, animal effects on ecosystems have been long discounted due in the system as compared to soil or soil microbial biomass or plant biomass. But there's been a growing body of work that has shown that animals can have really important impacts at the whole ecosystem level. And this was beautifully illustrated in a review paper led by Oz Schmitz, and on which Sean Leroux was also a contributing author. It came out in 2018 and coined my new favorite word, zoogeochemistry, is that animals can have even really significant impacts on carbon cycle through strong positive and negative feedback effects on trophic structure through kind of a variety of different potential pathways. And through this compilation of case studies that they presented, they show that animal effects can range from enhancing or reducing carbon storage anywhere from 10 to 1100% of that which would be happening without the animals on the landscape, with a median change of 40%. And these case studies really ranged a whole, gradient of taxa and animal body size, from stoneflies in streams up to musk ox and the Arctic mire. So it's not just the case of large animals driving these things or abundance of small animals. It can happen really across a pretty big gradient. And in the system, in fact, that we were just flying over together, and another paper by Oz Schmitz that he published in 2014, comparing the influence of animal effects on carbon storage with anthropogenic effects, he showed that the Serengeti wildebeest migration actually had an impact on the carbon footprint, which is shown here in brown, that was on the order of and even exceeded that due to all of the anthropogenic carbon emissions from fossil fuel carbon emissions in Africa. And this was one of 5 case studies that they highlighted in this paper in which the animal effects were on the same order of magnitude as those from fossil fuel carbon emissions. And in this system, this is largely driven by the grazing of wildebeest in the terrestrial ecosystem. And so grazing increases net primary production and thereby increases carbon fixation on the landscape. But it also decreases CO2 released from wildfires that are more abundant when grazing is low. And so by comparing when wildebeest numbers were low due to rinderpest versus modern day numbers when they're high, they could get an estimate of how much the wildebeest have influenced carbon cycling on this landscape. Animals can have really important effects on ecosystems through kind of 3, what I kind of think of as 3 different main mechanisms. There are trophic effects, so animals acting as either consumers or prey. Keystone predators are kind of a classic example of animals that can have an outsized impact on community structure, and a growing number of studies have now gone to those classic keystone species studies and shown that it in turn affects carbon dynamics in systems. Animals can have really strong physical effects on systems. So the classical concept of ecosystem engineer are animals that alter the abiotic environment in ways that then influence community processes. And these can translate into effects on carbon and biodiversity.
Oops. Sorry, everybody. Looks like maybe we lost Amanda. Get in touch with her and see if she can hop back on.
In the meantime, maybe Cole, you could ad lib or.
Sorry, Sean, what was that?
I was joking. I said, in the meantime, maybe you could just ad lib.
Just pick up where she left off. Hang tight, guys. We are emailing her and seeing. Ah, there she is. Up, you're muted there, Amanda.
Should I turn my video off?
I'm wondering if that was the issue. We could try it that way.
Okay, let me see. Are you guys seeing the right screen now?
Yes.
Okay. Yeah, I was seeing some like freezing happening.
Because I think your video screen was frozen for me, but I thought maybe that was just on my end. But
Okay.
yeah, maybe
let me turn my video off and see if that improves things. Sorry, I'm trying to get this back here. Okay. Well, we'll give this a try. If I lose you again, I'll come back.
Cool.
Or if you lose me. So okay, many ecosystems are structured, are largely influenced by resources that come in from external systems. And some really cool work by Leroux and Leroux formalize this idea. Well, there's a lot of different ways that these resource subsidies can influence recipient systems. And one of the ones that I think has been really informative for me is this idea of a subsidy hypothesis, which basically proposed that systems that are concave, because of their shape and position on the landscape, tend to receive much higher loads of resource subsidies of allochthonous inputs. So water bodies, and you all are in an area where you have deciduous leaf fall. I was seeing that, but kind of the classic subsidy is just leaves falling into streams, and you can think how many aquatic systems tend to be on the much higher recipient end of allochthonous inputs than other systems, and that this could be a possible explanation for, like, for example, the strength of trophic cascades in aquatic ecosystems. I am particularly interested in animal vectors of resource subsidies because they differ in a few really key ways from abiotic vectors. So although leaves are biotic when they're alive, once they've died, we can potentially classify them as an abiotic vector that's largely being transported by gravity, down into the streams or down the slope. But animal resource subsidies are unique in a few key ways. First, they can be very high in limiting nutrients. So we're often talking about animal excretion, urine, feces, or carcasses. And these tend to be very high in nitrogen and phosphorus and other micronutrients that are often limiting in ecosystems. Secondly, animals move. So they can move resources at faster rates along natural gradients or even against natural gradients. So, anadromous fishes swimming upstream are kind of a great example of that. And finally, animals aggregate in time and space. And this can lead to hot spots and hot moments of biogeochemical cycling that can have potentially disproportionately large impacts on both biogeochemical cycles and ecosystem function. But there's a large variability in the ecosystem effects of animal subsidies. So if we take kind of the most classic, I would say, well-studied example of animal resource subsidies, that is the anadromous Pacific salmon. So these are, fishes that spawn in fresh waters, spend the majority of their life accruing biomass in the marine system, and then return to freshwater systems to spawn again and die. And the classic story of salmon is that they then, through this process, fertilize the headwater streams that their young come out of. But actually, and this is just kind of a very partial review of a series of studies on Pacific salmon, but it really reflects the variability in results that you see in the literature. In some cases, they increase primary production. In some cases, they decrease it. In some cases, they have no net effect on it. So it can be actually difficult, even in a system that's as well known and seems as straightforward as salmon, to predict what effects animal subsidies may have on ecosystem processes. And I propose that this is because there's a lot of context dependency in the ecosystem effects of animal resource subsidies. It really depends on characteristics of the animal vector, like their mass or population size, characteristics of the donor system that they've come out of, like productivity dynamics or climatic shifts that have driven shifts in productivity. Those things together influence the potential subsidy coming into a system, but the effects of that subsidy are then translated through the lens of the recipient ecosystem. So physical and chemical characteristics like the size, a big fish in a small pond has a bigger potential effect than a small fish in a large pond. Also characteristics of the consumers, what kinds of trophic guilds there are represented, and how mobile potential consumers are in their ability to take advantage of pulsed resources. And altogether, these can kind of combine to influence ecosystems in a variety of ways. A lot of my research is in sub-Saharan Africa and Eastern Africa, and this is a really cool place. I was drawn to do research in this system specifically because there's a very large proportion of large-bodied wildlife still extant on the landscape. So Africa is the only continent with most of its megafauna still extant. And big animals tend to do big things. They eat a lot, they defecate a lot, they move a lot. And this enables us to kind of study the potential role of animal resource subsidies in a place where they're happening at a really large scale. So my research focuses in the Serengeti Mara ecosystem, and in particular in the Mara River, which runs from the mountains of Kenya through the Masai Mara National Reserve into the Serengeti National Park and then out into Lake Victoria. And it's the only permanent source of water in this ecosystem. So it's a really important resource for both the people and the wildlife that live in this region. And it's also a really cool place from an animal subsidy standpoint because there are two big potential sources or vectors moving resources from terrestrial and into aquatic ecosystems. So there's a lot of hippos, which eat grass at night. They leave the rivers to eat grass at night, and then they return during the day where they spend their time kind of aggregating in these hippo pools and doing their business and kind of lounging along one another. And when they do that, they're transporting all that grass back into the river system. And the Mara Serengeti is also host to the largest remaining overland migration in the world of about 1.3 million wildebeest. And these animals migrate from the breeding grounds down here, which is potentially more fertile soils, but drier. And in the drier season, when all of these seasonal rivers start to dry up, they migrate up to the Masai Mara, where it's much wetter. And this is where they cross the Mara River multiple times as they move kind of between these dry season feeding grounds. So they're usually in the Mara from July to October, and they'll cross the river a lot of times. And this is a little bit of a deceptive figure because they're not moving as a herd of 1.3 million. It's kind of there's altogether that many, but they can be quite distributed in space. So you'll have groups of anywhere from a few hundred to a few thousand to 10,000 crossing the river at various points in time throughout the time that they're up in this region. And these river crossings are, really iconic. They're probably one of the more photographed natural phenomena that happen. And occasional mortality will ensue due to a crocodile maybe taking, crocodiles will hunt at these crossing sites and take individuals. But they're also just very treacherous. And so you can see here wildebeest scrambling to get up these banks. And the more wildebeest that cross, the bigger the herd is, the more slippery the banks get and the harder and harder it gets for them to get out. And they start to pile up against one another at the far side of the bank. So you can see here some hippos kind of grouchily sharing their home pool with this herd that has decided to cross through, and some big crocs over here kind of biding their time. And much of the work has kind of, much of the, I would say, focus on these crossings has been on these individual instances of mortality that occurred due to crocodile attacks or maybe individuals breaking a leg. So this zebra escapes, but the others must be feeling a bit wary as they head in. But what happens sometimes is that if the water is high enough, if the crossing site is bad enough, wildebeest can pile up on the far bank. And if they aren't able to get out of the river and they start having to tread water, they can't do that very long. And they'll eventually start to drown. And this can result in these mass drownings of hundreds or thousands of individuals. And this had been kind of noted. And there had been some work done by paleoecologists looking at these specific sites as kind of a way to understand bone bed formation development in prehistory. But they hadn't been really regularly documented and their ecological impacts hadn't been studied. So when we first got to the Mara and started working there on a water management project back in 2008 and came to the river, one day and saw this huge number of carcasses along the river, I just thought, this is incredible. This is like salmon in the Pacific Northwest, but happening for ungulates here. And how has nobody really looked at this? So ensued my research working in this system and studying these two vectors. And so I'm going to talk about their inputs and their ecosystem effects along this gradient that we work in. And for the purposes of the talk, I'm going to refer to these three main study sites as none. So this is a site, there's really no large wildlife inputs upstream here. This is mostly dominated by human inputs and small scale agriculture. So it's a little bit of like our reference site. Once you get downstream, there's about 2,000 hippos between here and this hippo site. So there's about 2,000 hippos upstream of here, and then another 2,000 down to this hippo wildebeest site. And in between here is when the drownings happen during parts of the year, just these few months from like maybe August to October. And so we are able to kind of to a degree parse these different inputs in time and space to understand their different ecological effects. So the overarching question I'm going to address today is, how do hippo and wildebeest subsidies influence ecosystem dynamics in the Mara River? And this is kind of a framework I'm going to use to walk through this. So our first objective was just to quantify how much hippos are bringing into the system and quantify how much wildebeest are bringing into the system. And I thought that this would be easier than it turned out to be, but it proved to be more complex and more interesting than we expected. Then our understanding of how these different inputs together influence ecosystem function in the river and how that translates into food web effects. And you'll see kind of a current running through this whole theme is the influence of discharge, which is this background level of environmental context In river systems, discharge is thought of as like a master variable. It influences all components of a river ecosystem. And so it's a really good lens through which to think about how environmental context influences the translation of these subsidies. So starting with hippos, hippos are overall have declined really substantially, although they've been listed by as vulnerable by the IUCN, but in the last assessment, they did say that their populations had started to stabilize, but there's been a pretty big decline in their population. You know, they don't play well with others, so they're not always popular in certain regions. But in the Mara, they've done quite well. They probably were artificially low numbers in the 50s due to hunting or poaching, but since the region has become more protected for wildlife, there's been a 1,500% increase in their population. So it appears to be fairly steady over the last 10 years or so. There's about 4,000 hippos in the Kenyan portion of the Mara. And we were trying to get our head around how to quantify what hippos bring into a river system. And aquatic ecologists actually do this a lot for fish and bugs. They measure excretion by taking a bag of clean water, measuring the nutrient contents in it, putting a fish or a bug in for a certain period of time, and then measuring the nutrients after. And that difference gives you how much that animal excreted over that period of time. And I was kind of bemoaning to a colleague that we couldn't do that with a hippo, and he was from Milwaukee and told me about the Milwaukee County Zoo exhibit, where there's basically a hippo pool with 3 hippos, and every day the zoo staff clean it out and fill it with clean water and let the hippos come in. And they were more than happy to have us come and kind of shovel hippo dung out, carefully measure it each day for a couple days in a row to get an estimate of how much hippos are excreting and ingesting into water bodies over a 24-hour period. And just to give you kind of a view of what that looks like. Here's cleaning the pool, filling the pool with clean water. Here's our 3 resident hippos hanging out. There's one in the back there. And after less than 24 hours, you can see a pretty profound impact on water quality. So we used data from that to parse excretion and ingestion. We used data from the literature to use like a metabolic approach to estimate intake, stoichiometry of food from the field. So we kind of brought together a range of different field and literature and experimental estimates to get an overall estimate of how much hippos are transporting into the Mara River in particular. And we estimate that they contribute about 13,000 tons of feces every year. So that's about 3,000 tons of dry mass, organic material, every year to the river. And a lot of that is carbon, about 1,300 tons of carbon that they're dumping in every year. That is a lot, but it's really relevant to ask, is it a lot compared to what is coming in from other sources, right? That's the key to understanding recipient system, the lens of the recipient system. But when we compare it to what's coming in that none site with no wildlife upstream, see that hippos are increasing the amount of particulate carbon in the river by 670%. Their inputs comprise like a quarter of the nitrogen and almost a third of the phosphorus that's coming in from upstream sources. So even compared to whole watershed loading, these hippo inputs are really a substantial part of the nutrient and carbon budget in this system. So what happens when hippo dung enters the river? So we used, again, another kind of adopted a method that's often used for leaf litter to look at decomposition rates, and we made litter bags with hippo dung and measured decomposition rates over time, and also looked at the stoichiometry of feces and the urine. We were able to parse the two from the zoo work. And we found that, actually what we have thought of as one input from hippos is really 2. Hippos are contributing dung, and that's like 88% carbon. And it takes about 80 days to decompose in the river system. And because it's particulate, a lot of that settles out on the river bottom and then just slowly decomposes over time. But hippos are also contributing much of the nitrogen and phosphorus through urine, which is soluble and can readily move downstream. So there's a lot of complexity in the inputs coming in here. And these inputs are aggregated in space similar to hippos. So this is a map of hippo aggregations in this region of the Mara, but translated into carbon, nitrogen, and phosphorus loading. And you can see that there are indeed these overall hippo pools are kind of hotspots of loading, but even among them, they really range in the amount of total, like carbon can range anywhere from less than 4 to, over 100 kilograms per hippo pool per day. These hippo pools and their inputs then are hotspots of biogeochemical cycling. So in work done by my research partner and husband, Chris Dutton, he showed that, so he used this cool robotic boat. It's tricky to work in hippo pools. They're territorial, they can be very dangerous, and they really don't like other things coming into their pools. So this was work we did with a company called Platypus that helped design this. We were using it here as a remote control boat. It can be driven robotically as well, with a low-cost water quality sensors that could kind of create a 3D interpolation of hippo pools. And you can see the difference in stratification that happens in conductivity in a pool with no hippos versus a pool with high density of hippos. So the bottom of these high density hippo pools is, really heavily stratified with really high levels of conductivity and also really low levels of dissolved oxygen. And when these high subsidy hippo pools are flushed out by high flows, you can see a record of time here of discharge. Every time discharge in this river system peaks and all these hippo pools are flushed out, we see these complete crashes in dissolved oxygen that happens downstream. These hypoxic floods are extremely unusual. We're not familiar with them having been really shown in any other river systems, although we think that's probably due to lack of monitoring. I imagine that they do happen in other tropical river systems in some places, but it's really unusual for an increase in flow levels that usually re-aerates a river to actually cause a hypoxic flood. But we think it's due to the scouring of all of the reduced compounds in those high subsidy pools. And these can cause huge fish kills that happen, hundreds and thousands of fish lined up along the banks of the river. And water residence time is a really big driver of those biogeochemical state changes. So you can see this is just a PCA of biogeochemical variables, and the hippo pools really group by concentration of hippo subsidy. And what's really happening here, what this really illustrates, is a shift between oxic and anoxic states. So low subsidy hippo pools and pools that don't have hippos remain in an oxic state, whereas ones that are medium or high subsidy remain in an anoxic state. And this has pretty big impacts on biogeochemical cycling. So we get high levels of reduced compounds and high levels of like greenhouse gas emissions from these hippo pools over here that are in an anoxic state, where lots of carbon is decomposing under anoxic conditions. Whereas hippo pools over here that are in an oxic state with high levels of nutrients can stimulate production. So the quantity of loading has an effect, but the really big driver here that determines how these parse in space is water residence time. So again, it comes back to discharge as this kind of environmental driver impacting how these inputs are translated into the system. So that's a bit of what our journey towards understanding the role of hippos and influencing this river ecosystem. They're contributing 13,000 tons of feces to the river every year. And this is really these kind of two different forms of subsidy input, both a particulate input that's high in carbon and a soluble input that's high in nitrogen and phosphorus. Okay, so what about wildebeest? Well, similar in some ways to the hippos, globally, many large migrations have declined or been extirpated altogether. And the Serengeti Mara ecosystem is yet another outlier in this regard, in that the wildebeest are actually doing quite well. Their population had been knocked down quite a bit by rinderpest in the early part of the 1900s, but they've generally recovered since the 70s or 80s and remained at this fairly stable population size of about 1.3 million since. So there's a lot of wildebeest in the river system, and our goal though was to understand how many might be entering the river through these mass drownings. Was this just kind of a freak event that we happen to observe once or twice, or was this an annual occurrence? And so we used historical records from safari lodges and resource managers in the system going back to 2001. And we've continued to keep up surveys. We did detailed surveys in 2011 to 2015, where we really quantified every drowning event to the best of our ability. But we've continued to keep up overall survey, it's in the last 5 years. And we can say now that there have been mass drownings that we've recorded or have been recorded in 18 of those 20 years. And during the years where we've been able to count them in high detail and really track them closely, we counted 23 drowning events over those 5 years that range from like 100 to 5,000 individuals per drowning. So a mean of over 6,000 carcasses per year going into the river system. Now, this is really nothing compared to the whole population of the Serengeti wildebeest, right? It's less than 0.5%. And so it doesn't have a impact really on the population of the wildebeest per se, but it's a huge input to this river that has a mean discharge of about 12 cubic meters per second. So, okay, 6,000 wildebeest, 6,250. What does that really mean though? What is a wildebeest bringing into a system? So we pulled a few fresh carcasses out of the river. They're quite heavy. About 175 kilograms is a mean mass of a wildebeest. And we basically dissected them into their various components and then measured elemental stoichiometry of those so that we could rebuild a wildebeest essentially and understand what was coming in. And one really interesting observation of that was just that, first of all, wildebeest are very lean. There's essentially no meat and no fat on these fellows. But they also, a lot of their, the mass of their carcass is bone. And this isn't surprising if you look at kind of predictions of stoichiometry with body mass, but it was hard to find data on this, which is why we had to go kind of collect our own. And this for me really crystallized the importance of body size and understanding the role of carcasses in a system. Because while altogether wildebeest are bringing in 1,100 tons of biomass, when you start to look at the constituents of that biomass, a lot of that is actually in phosphorus, and maybe more than one might expect. So again, comparing this to hippo, or comparing this to the watershed loading that's coming in from upstream during the period of time. So this is scaled only for the period of time when carcasses may be in the system. These inputs are a lot compared to the whole system. About 78% of dissolved carbon, a third of the nitrogen, and almost 200% of the phosphorus coming from upstream that carcasses are bringing into the system. We tried to do the same thing with the litter bags for carcasses that we did with hippos, but after about 1 day, we realized that the Nile monitors and Nile crocodiles had just thought we put out a bunch of party favor bags for them, and they had plucked each of the bags off these long chains that had carcass material in them. They were actually remarkably good at taking only those bags. And so we got a welded cage and redeployed litter bags with carcass material inside this welded cage. And we kept it out in the system. This is a scaled in or zoomed in view of the graph just to illustrate trends better, but we kept it in for about 8 months, which was as long as we could maintain it. And you can see that the soft material, the soft tissue decomposed really pretty quickly. Within, we're looking at decay rates, it was gone within, 30 to 45 days. But the bone tissue broke down much, much more slowly. So at the end of 8 months, it was very almost negligible, the amount of mass that was lost. And so we estimate, we estimate that these are breaking down on these decadal scales. So, potentially, now these are protected inside of a welded cage. So this may be on the long end of decomposition rates, but quite long. So similar to hippos, wildebeest are really bringing in almost 2 different forms of subsidy. 95% of that phosphorus is in their bones. Their bones have a very different stoichiometry compared to the soft tissue, whereas most of the carbon and nitrogen is in that soft tissue that is, relatively quickly mineralized and potentially moved downstream. Also similar to hippos, wildebeest carcass aggregations act as kind of hotspots of this nutrient loading, and because they're kind of both aggregated by drowning event just downstream of crossing sites, we are able to kind of put barriers at the, treat it as a box a little bit and approach this with a classic ecosystem measure of understanding fluxes in and out of this box. And so we did this we kind of compiled the data for all the drownings that we were able to measure of this time period to try to understand the fate of these nutrient inputs in the system. So this is the stoichiometry of the initial loading for a mean annual inputs of wildebeest carcasses. And a lot of those nutrients are retained in bone. So it is 95% of the phosphorus, but it's also a quarter of the nitrogen and about a third of the carbon is potentially just retained in this system over very long time scales. And because there's really high input rates divided by really low decay rates, what that gives you is a very high estimate of standing stock. So we estimate now that there's, the standing stock of bones in the river is, I think, 0.6 kilograms per meter squared. A fair amount of that nutrient is just transported downstream. The system becomes saturated with high amounts of nutrients, and so we see just big peaks in dissolved nutrient concentrations moving downstream, which is likely assimilated in further reaches, but could be transported quite far. So our nutrient uptake length estimates are in the order of 10 to 20 kilometers that some of these moles of nutrients may be transported before being assimilated. Some of it is facilitating biofilm growth. So we could measure nutrient uptake, but it was just at very, very low rates relative to what was coming in, probably because these inputs are coming into a place that is already impacted by about 4,000 hippos. So it's not a low nutrient system that they're coming into at all. And then fascinatingly, a lot of them are actually being consumed by vultures and then transported back out to the system. So back out to the terrestrial system. So vultures are consuming portions of the soft material and leaving bones behind in the river system and transporting, close to nearly 10% of nitrogen and carbon, potentially moving back into the terrestrial system. Overall, we can account for about half of the carbon and the nitrogen. Which is, I'm told, not bad for an ecosystem budget approach. It's hard to track for all these things. And we know we're missing. We aren't, in this estimate, we weren't able to account for in-stream consumption because that's a much more difficult thing to measure, but something that we're currently working on doing. And we also aren't accounting for atmospheric loss. So all of those little biogeochemical hotspots are probably also anoxic during certain times and losing a lot of CO2 and nitrogen gas to the atmosphere. So when we compare these two sources of input now, we see that wildebeest are bringing in less than a tenth of what hippos are bringing in terms of raw biomass, but it varies quite differently in its potential fate. So what stays behind in the system is really high phosphorus, whereas what's more soluble and maybe more quickly mineralized and assimilated is the carbon and nitrogen-rich soft tissue. So how do these two inputs together influence dynamics of the river ecosystem? And I think it's really helpful to think about this through a stoichiometric lens. So if you're not familiar with thinking about resource stoichiometry, it's basically just the ratio of carbon to nitrogen to phosphorus or any element of interest. And there's this classical concept known as the Redfield ratio that posits that much living material, although this was developed for marine phytoplankton, has a stoichiometric ratio of 103 carbon to 16 nitrogen to 1 phosphorus. So I've scaled all this to 1 phosphorus. So carbon to nitrogen ratio in much biological material scales around this ratio. And if something has more carbon than nutrients, it's kind of considered lower resource quality. There's a lot of processing that has to be done, digestive processing, to get to the good stuff. If something has less carbon and more nitrogen, relatively speaking, then it's a higher quality resource. You don't have to, think of this as eating the whole pizza and the pizza box, whereas this is kind of just focusing on the cheese and the pepperoni. So we can see that the resource subsidies that we're looking at here really range quite widely in their stoichiometry. And you can use that to make predictions about how these resources might be assimilated in the river ecosystem. So much of the hippo inputs are through egestion or feces, and this is organic material. And that is high in carbon. So it's likely to mostly stimulate microbial respiration in the system and maybe be directly consumed by herbivores and detritivores. Carcasses are mostly bringing in organic material as well, but it's much higher, relatively speaking, in nitrogen and phosphorus. So a lot of this could stimulate increase in primary production that may move up the food chain through the green, kind of through the green food chain, the algal food chain, as opposed to the detrital food chain. And so this is how we might hypothesize that these different resources could be assimilated into the river food web. So a few metrics that we looked at to try to measure this. First, we looked at nutrient levels over time. And we see that, as you would expect, both nitrogen and carbon and phosphorus all go up as we go downstream from no to hippo wildebeest sites. And when we measure this, we measure nutrient limitation. This is an approach used in aquatic ecosystems where you grow biofilm on various cups that either are amended with nutrients or not. And if you get more nutrient growth on the cups that had nutrients added, then you can hypothesize that there was nutrient limitation happening in the system. And so we did measure nutrient limitation of autotrophs and nutrient and phosphorus co-limitation and carbon limitation of heterotrophs up in this upper site. Even though it's not a particularly low nutrient site, there's very little organic material coming in here, but no evidence of nutrient limitation going downstream. So it appears that new wildlife inputs have reduced nutrient and carbon limitation in the system. How does that scale to whole measures of primary production in the system? Well, we looked at this through two ways. One, through an experimental approach using these artificial streams. This is a portable artificial stream array that we built and used in Kenya. And we have one that we're just built here at UF that's a bit more formal looking maybe than this one. But so this is gross primary production. Just to compare to the ecological site, I'm just going to show you, or the ecological data, I'm going to show you GPP at the site with no wildlife inputs. Hippo inputs don't have a big impact on primary production, but once you have hippo and wildebeest together, you get a big increase in primary production. And this is what we might have hypothesized, right? Higher levels of nutrients coming in with these carcasses could stimulate primary production. Whereas when you look at respiration, it's really the hippo inputs that are causing the increase in microbial respiration. And this is important, this balance of production to respiration, for a number of reasons, but one of which is it tells us about the ability of the stream to act as a carbon store or source of carbon to the atmosphere. So the more production that's happening, the more the river is storing carbon and translating it into potentially the river food web. The more respiration that's happening, the more the river is acting as a source of carbon to the atmosphere. But this is really different than the data we got when we looked at the river. And we did this using estimates of whole river metabolism from diel oxygen curves. And we see that gross primary production here increases with hippo inputs, but decreases where we had both hippo and wildebeest inputs. And if you compare that lowermost site to when there are carcasses present versus when there are no carcasses present, there's really no difference. So whether or not there are 5,000 wildebeest carcasses in the river doesn't appear to influence primary production. And as surprisingly, or more surprisingly, respiration does increase as well with hippo inputs, but there's no real input of respiration, no change in respiration at the lowermost site. And when you have carcasses present, respiration actually decreases a bit, which is a bit surprising. So what might be driving that? And I think the hypotheses basically fall into two camps. And one is wildebeest don't have a big effect because the hippo effect is so profound in the system that it's kind of a drop in the bucket. It's less than 1/10 of the biomass on top of what's already being processed and assimilated. The other potential hypothesis is that wildebeest impacts always matter at that lowermost site because the bones are always there. So there is no lowermost site with or without carcasses. The bones are there on these decadal timescales. So that site is fundamentally impacted by wildebeest inputs. And for whatever reason, that is leading to this or potentially contributing to this pattern with lower levels of primary production. So this kind of surprising non-linear impact of wildlife inputs on primary production. And how might that be translated to the food web? Well, this is work that we're kind of in the middle of. So to show you some of where this is at right now, we've done some initial surveys of insect and fish biomass in the system and started using stable isotopes to look at assimilation. And just with kind of an initial survey of biomass, we see that aquatic insects and fish biomass increase as you go from upstream to downstream. So there seems to be just more bugs and fish in the river. And they seem to be using and assimilating these wildlife inputs. So here is fish tissue assimilation for one of the most common fishes in the river, Labeo victorianus. And you can see that hippo feces here is about 35% of their assimilated tissue. And when carcasses are present in the river, carcasses actually comprise an even higher proportion of their assimilated tissue. So whereas we often think of things as preferentially feeding on algae when possible, in this system, wildlife inputs provide a really important resource during the wet season. And what was really interesting was we thought, well, maybe during the dry season, we would see a bigger algal signature, but we actually see the opposite. Hippo feces becomes even more important. And I think this is because it settles on the bottom of the river. As I mentioned, it's a particulate resource and it's settling on the bottom, potentially shading out algal resources, and is comprising, upwards of 80% of the assimilated tissue of fishes during the season. The other really surprising and cool finding was bone biofilm is still showing up as a resource. So carcasses in this sampling period have been gone for several months. The soft tissue decomposed 3-4 months ago, but the bones persist. And it turns out that bones grow biofilm, and that biofilm looks isotopically different than the biofilm that grows on rocks. And it looks functionally different as well. So the chlorophyll A and ash-free dry mass, so like the amount of organic material in biofilm growing on rocks, on bones, is 3 to 5 times higher than that growing on rocks. So even though there may be less bones in the system than rocks at this site, there's still a lot of them. Remember, we estimate that the standing stock is quite high, up to 0.6 kilograms per square meter. And the biofilm may be actually providing a preferential resource. So wildebeest continue to influence the food web of the system even months after drownings have occurred. So this is this struck us at the time as a very interesting pattern. We see a non-linear effect of wildlife subsidies on primary production. It increases at intermediate levels, but it decreases at very high levels. This is data shown from the river system, but from ceramic tiles. And we see what looks like it's probably going to be, this isn't well replicated at this point here, but an increase in insect density as we go downstream. And so this has formed the basis of the current work that we're working on in the system, which is quantifying how the magnitude of subsidies might be influencing the trophic basis of production and the strength of trophic cascades across this gradient. So if you remember what I said about the subsidy hypothesis, the idea that increased amounts of subsidies may drive and strengthen drive the increased strength of trophic cascades in certain systems. We hypothesize that where there are no wildlife subsidies, algae is an important important food resource for aquatic insects and fishes. But at high levels of these resource subsidy loading, of wildlife subsidy loading, fish and aquatic insects populations may grow to such a size that they're providing a very top-down force on the algal populations. And potentially that is what's driving this decrease in primary production. So if you remember this figure that I showed you here with our various hypotheses about how these different resources may influence ecosystem assimilation, basically we hypothesize that this high level of input to primary consumers here may then lead to a top-down effect on primary production. So we're still in the kind of early to middle stages of this work, not at all facilitated by the global pandemic that's been happening, but we've had this amazing postdoc, Therese Frauendorf, who has been leading the research and managed to, by assembling and training a really outstanding field team of local researchers, the research has been able to continue over the last year. So the data is kind of still coming in, but thus far what we see is that hippo inputs along this gradient of wildlife inputs, we see a big increase in the density and the diversity even of invertebrate taxa. It's not consistent across which taxa are facilitated by hippo feces and which are not, but overall an increase in diversity. And what's interesting is that here we actually see a fairly consistent number of insects per gram of hippo dung. It's about 30 to 50 invertebrates per gram of hippo dung. So that doesn't change, but what does change is the sheer quantity of hippo dung. So the amount of hippo dung coming into the system grows, and with that, so does the density of macroinvertebrates. So we are seeing a really big increase in magnitude of primary consumers as we go downstream. And so we did an experiment to look at to what degree this increased magnitude of subsidies could influence top-down grazing. And this is a grazing exclusion experiment that we ran in the Mara, where we basically used some pre-colonized and some not pre-colonized discs. See that, you can see that here. And in the cups below, much like with that nutrient limitation experiment, we have some that are controls. We have some that are amended with malathion, which is toxic to invertebrates, but at these levels will just basically cause grazing exclusion by macroinvertebrates. And then half of all of the arrays, we put out 10 arrays at each site, and half of them were encircled with a copper wire that was hooked up to like an electric fence charger. So electric current that would reduce fish grazing. That's kind of set for body size that might be effective for fish grazing. And set up an effectorial design. So we could look at insect and fish grazing effects along this site, this transect of inputs. And just a kind of an initial look at these data, because this is something we're still working on analyzing, writing up, but we do see, so just to kind of orient you, this is data for the pre-colonized discs only that I'm showing you, although we saw the same pattern with the non-pre-colonized discs. And where malathion was added and basically reduced invertebrate grazing, we see higher levels of chlorophyll A. So the green bars are higher than the orange ones. And in the electric exclusions, we see overall higher levels. This is at sites A through E, and here's at sites A through E. So we see overall higher levels where the electric exclusion was applied as compared to the areas without electric exclusion. So we're seeing a significant effect of both insect grazing through malathion exclusion and fish grazing, and that varies by site and appearing to get stronger as you go downstream, although maybe not linear. Certainly a lot of variability in these data, but it kind of supports this idea. And this is part of a bigger project, as I mentioned, where we're putting this in context with secondary production estimates and estimates of the trophic basis of production to hopefully flesh this story out a bit more. So just to wrap things up, I see that I've managed to take up the whole time, so I apologize for that. But to wrap things up, a few points I want to leave you with. First, animals can influence elemental cycling at the ecosystem scale. This is a figure of work that we did with colleagues studying silica, showing that hippos can influence 30 to 75% of biogenic and total silica in the system. We saw similar numbers with carbon and nitrogen. And my guess is you would see this for a number of other elements that you might look at. And we saw similar numbers for hippos. So even compared to whole ecosystem levels, wildlife can have very large impacts on biogeochemistry. Secondly, those impacts can be highly context dependent. So as we saw with the wildebeest, you would think 5,000 carcasses rotting in a river would have a huge impact on river ecosystem production. But when they come into a system that's already heavily shaped by hippo inputs, and in which their consumers, primary consumers of carcasses, are vultures that are much more comfortable foraging on the land than in water, that mediates how those carcass inputs are translated into river ecosystem impacts. And finally, animal migrations, this is a cool place to study it because there are lots of large wildlife on the landscape. But I think it's important to think about the fact that animal migrations may have played an important role in other river ecosystems around the world historically. So just some North American context, there were 30 to 60 million bison in the US in the late 1700s. And they crossed many large rivers of the Great Plains. If you estimate that they drowned at a similar rate as we see in the Serengeti wildebeest migration, that would have been of just 0.5% of the herd. That would be an input of 150 to 300,000 carcasses per year in the rivers of the Great Plains. And in fact, there are historical estimates of this from some various trappers, including one who very studiously counted every carcass of a huge bison drowning he documented on the Assiniboine River in the late 1700s, and counted about over 7,000 bison carcasses, from which we can estimate that contributed about 43 tons of phosphorus, about 50% of what we now know is like an annual load of phosphorus in that river. And so, although we don't have much data on these historical drowning events, I think that they were probably much more common across the landscape when large migrations of animals were more common. And so I think, a lot of the work done by, this is a figure from a paper done by Chris Dowdy titled Global Nutrient Transport in the Time of Giants. And I think it's a great place to end, because what he really illustrates here is the role that, the idea that the ecosystems that we study are largely shaped by the loss of these animals that used to play important roles in nutrient transport. And so I think of this quote by Luna Leopold: "Rivers are the gutters down which flow the ruins of continents." Nutrients are on a constant one-way journey from mountaintops to the bottom of the ocean. That's the way that they move. And there's not a lot of things that move them back upstream, but animal movements are what can do that. Whales forage in deep waters and through fecal blooms bring those nutrients back up to the photic zone. Seabirds feed on the fishes that feed on those fecal blooms and the algal blooms that they support, and bring those nutrients back into roosting sites. Anadromous fishes feed on algal blooms and bring those nutrients back up into headwater streams. Animals come and feed on those fish carcasses and bring those laterally inland. So animals are the things that move resources back. And in the absence of those marine mammals and seabirds and anadromous fishes, this paper, they estimate that the movement of nutrients away from hotspots has declined to 6% of past values. So just a reminder and a reason, I think, for thinking and studying systems where the animals still do persist is that their impacts have the potential to be really enormous. And that even in systems where animals no longer persist, what we're looking at is a bit of a shadow of what may have once been. So with that, I want to take just a moment to acknowledge the many collaborators here and as well in Kenya who have been just a huge part of this project throughout. My advisors, Emma Rossi and David Post, and my partner, Chris Dutton, and a number of people who come to the field with us, and our colleagues at National Museums of Kenya, as well as a whole bunch of awesome REU students who have worked with us over the years. So thank you all so much for being here, for inviting me. And I know it's at time, but I'll be happy to stick around if anyone has any questions.
Great. Thanks, Amanda. That was an amazing talk. Really enjoyed that. If people have questions, if they just want to type them in the chat box, and then we can just go in order of how they appear in there.
Cool. Thanks, Paul.
I guess I'll just start with the question, and it might be kind of weird, but how did you actually parse out feces from urine in an isolated hippo pool?
Yeah, we knew what they were eating, and so we knew kind of like what should come in and what should come out, and then we took dung that we collected where they defecated in their cages, and we did leaching experiments to estimate like what proportion of what was dissolved in the pool was coming from the dung versus what was dissolved and maybe coming from other sources. And we drained the pool really slowly and shoveled all of the dung out into wheelbarrows and weighed it. So we basically were able to get all of the particulates, and we weighed it and accounted for it. We accounted for all the leachate from the particulates, and then what was left over was what we estimated was the urine.
Cool.
Yeah. Thanks. I see Yolanda said.
Yeah, Yolanda, if you want to go for it and ask your question.
Sure. Yeah. Thanks, Amanda. That was great talk. I missed just a tiny bit of it, but I think I got the cool bits. I was just curious, the whole idea of the bones sort of being this sort of permanent source for phosphorus is really interesting. I wonder if you've thought about in your artificial systems of trying to sort of have them be absent, because it would be clearly quite daunting to go and dredge the river and take them out. But it would seem to me to be kind of an interesting experiment to see what effect that might have on some of those nonlinear sort of productivity relationships you observe.
That's a great idea. So the work that I showed with this artificial stream experiment we did do with just carcass material. But we did an experiment where we had like 2 streams left over once we did all our treatments. So we put only bones in those. It wasn't like a fully designed experiment, but it did give us some really cool data about the fact that they were just bones we picked up out of the river system. Like, I don't know how old they were, but they weren't fresh. And we saw a huge spike in phosphorus and a huge spike in chlorophyll A in the first week after we put those bones in, that then kind of declined. So that was really cool. And I do think, I mean, I'd love to, it's work that kind of needs to be done there with, if we want to tie it specifically to wildebeest, but we've thought about doing it here too. Because I also find the bone story to be just kind of fascinating. And it was also totally surprising. Like, I just hadn't thought about that at all until we got into it. So I think there's more.
And I wonder, just kind of as a follow-up, if the sort of on some of the questions around sort of productivity, given that they are providing this sort of substrate for biofilm, but presumably the phosphate is coming from sort of slowly leaching from the bones. There seems to be kind of this confounding sort of input there, that it's the bone material, the elements sort of leaching from the bones, but then also this substrate for all this great biofilm that's going to contribute other things. And I wonder if there's a way to tease that apart too.
Yeah, that's a really good idea. And we've been thinking about like how to quantify substrate, like what substrate bone offers, because people have, there's particular algal, taxa that only grow on, turtle shells or, mussel shells. This has kind of been shown that just adding a hard substrate itself can be a resource. And interestingly, our Therese thinks her current hypothesis is actually that the hippo dung, maybe one of its most important roles may be adding structure as well, that the hippo dung itself is a structure. And so we've talked about maybe using like Easter grass, that kind of like plasticky grass, some kind of, basically how we could start to try to parse structure versus nutrient addition, from these different drivers. So I think you're definitely right.
That's neat. Thanks.
Thank you.
Cool.
Yeah.
Me? Did you just call me?
Yeah.
Kind of broke. Okay. Yeah. Thanks again for the great talk, Amanda. That was super interesting. I guess my question is, so I think it's like really striking that like these mass drownings rely on like that number of animals crossing the river at the same time. Do you think there's some like critical mass that makes these drownings happen? And like, is there potential? I guess I don't know much about wildebeest population density, but is there potential for that to be in peril in the future if population size changes in density, et cetera?
Yeah, so it's a great question. And I thought like a couple of years ago, like, oh, we should write a paper about what causes drownings, because that's probably pretty straightforward. Several years later, it's really not. It's been complicated to get a really good handle on that, because it seems to be these multiple factors that kind of all need to happen together. And it's really difficult to, first of all, just get data on the crossing, like all the crossings, like when they happen and when they don't happen, because that's kind of what we would really need. So we have put concerted effort into this during various field seasons, and I think we're getting close to having a good data set that we can try to parse these things out. But it's like, you really kind of need multiple crossings at multiple sites at multiple flow levels with multiple herd sizes to kind of have a good handle on that. So I don't know. I mean, the wildebeest population does not as of yet show a risk of declining, but not to say it won't, it's particularly if they build a highway across the Serengeti, which they proposed. But I think what is more likely to stop potentially drownings in the system are maybe two things. One is the system with climate change is just becoming much more unpredictable in terms of seasonal rainfall. And wildebeest in general, they're so tied to the rains. They seem to be spending less and less time in the Mara region. And we've just started talking with some folks in Scotland who track the wildebeest herd. And they were looking at their crossing, because they can look at crossing data with their collars. And they've seen a decline in number of crossings over the last like 5 years or so, which is really interesting. And I think that probably reflects just the lower amount of time they're spending in this region. So I think it's possible that they will just kind of not spend as much time in the region. The other thing is that the river is really something I didn't talk at all about today, but the river is a really important water resource for lots of different users, and overall flows have been declining in the river, particularly during the dry seasons. And climate change impacts are probably partly responsible, increased severity and frequency of droughts, as well as increasing extraction. And there's, of course, discussion about building dams on the river, because that's what people discuss doing to rivers. So, I think that at very low flows, and we've seen this in the river, in the years when those 2 years when no drownings happened, it was because the wildebeest could just stand in the river while they waited to get out. So that does seem to, there's like a bit of a threshold. It's like below 5 cubic meters per second, like no drownings happen. And then above it, it's actually pretty linear. Like there's not an obvious really big discharge effect, but there is a minimum flow below which they won't happen anymore. And I think that is probably one of those two things